This is an oak tree. It doesn't look much like one, but it is. A small acorn has the complete potential to become an 80-foot-high oak tree, because within the acorn is the blueprint for every cell of that oak tree. In the living process of growth, the cells of a seed are duplicated billions upon billions of times, guided unerringly by the blueprint of its genetic material, resulting in the transformation of that seed into a tree. And the essence of the growth process is the same for virtually all living organisms on Earth. Each living thing has its own special blueprint for its species. Now, the amazing thing is that the blueprint for every single living creature on Earth is composed of the same kind of molecule, which scientists know as DNA. The DNA is organized into tiny subunits called genes. Genes are responsible for producing the various traits of an organism. DNA does this by supplying the information needed by cells for the manufacture of the proteins that make up all living substance. In a sense, DNA is the alphabet of life. Just as there are few words in English that consist of only a single letter, most traits in an organism are the result of the interaction of many genes. For example, each book you see contains a different story. Yet every one of them is written in words made up of letters from the same alphabet. The number of meaningful stories that could be written using different combinations of the same letters is almost infinite. The different organisms we see around us are the different books of life that DNA has been used to write. Organisms grow by the duplication and division of the cells within them. Mitosis is the method by which nearly all such cell division occurs, and is the means by which the master plan, the DNA, is passed from cell to cell in a growing organism. If we watch mitosis in action, as in this single-celled animal, it is easily seen that the result is the formation of two identical cells from an original cell. In watching cells divide, we should try to be aware of their three-dimensional nature, as these scanning electron micrographs of mitosis illustrate. This healthy young salamander is undergoing rapid growth to maturity. One would expect its cells to be in a constant state of division. However, even in the salamander's most rapidly growing tissues, the cells divide only about twice a day, and most cells divide even more slowly. For the greatest portion of their life cycle, cells are in a state known as interphase. During interphase, the DNA found in the nucleus is distributed as an indistinct granular substance known as chromatin. While biologists used to think that interphase was a resting period for a cell between divisions, we now know that nothing is further from the truth. Although it isn't obvious, the cell during interphase is a dynamo of activity. During this time, the cell is engaged in duplicating all of the structures and materials that the new cells will need following division. Among these, perhaps the most important, is DNA. The crucial process of DNA replication occurs near the middle of the interphase period. It is important to remember that chromosomal DNA is replicated only during interphase, and that it is a process that does not occur during any other stage of a cell's life cycle. Further, this replication of DNA must be absolutely exact, so that the two identical copies of the genetic material can be passed on without modification to the two new cells that will be created in the coming division. After replication is completed, the DNA, shown here as a simple chain, begins to coil upon itself. And while proteins are associated with this coiling, they are not shown so that we may watch more closely what is happening to the DNA. As we move through the coiled DNA molecule, we reach a point called the centromere, where the molecule is joined to its exact replicate. Together, these two DNA molecules form a chromosome. The appearance of chromosomes marks the visible beginning of the division process. As the chromosomes appear, the nuclear membrane disappears. In this salamander cell, the chromosomes now migrate to the center of the cell, aligning themselves along its equator. The centromere of each chromosome splits, and each half of the original chromosome now becomes a chromosome containing only one molecule of DNA. The halves move to opposite sides of the cell as the cell membrane pinches in, forming two new cells. Each of these new cells now forms a nucleus containing the same number of DNA molecules, identical in structure to those in the original cell. The division we have just seen actually took about 20 minutes. The two new cells formed in mitosis now enter interphase and will not be ready to begin division again for another 24 hours. However, not all cells are destined to keep dividing over and over again. Human nerve cells stop dividing shortly before we are born, and if they are lost, can never be replaced. Other kinds of cells will only divide under special circumstances. Liver cells, for example, never divide in the adult organism unless the liver has been damaged, and cell division is required to repair the organ. During interphase, the cell duplicates its DNA. The chromosomes become visible when the DNA coils. The chromosomes align themselves in the center of the cell. The chromosomes then split and move to opposite sides of the cell. The single division of mitosis results in two new cells, both with DNA identical in composition and amount to the original cell. Meiosis is another kind of cell division that makes possible the fundamental life process of sexual reproduction. In sexual reproduction, a cell from a male of the species is combined with a cell from the female of the species, resulting in a single cell, a zygote, which can grow into a complete organism similar to its parents. Meiosis accomplishes this by creating a new kind of cell, sex cells. Sex cells have only half the DNA found in the other body cells of the organism. For example, humans have 46 chromosomes. 23 of these come from the mother and 23 from the father. Meiosis makes this possible. Meiosis reduces the number of chromosomes in half, in this case from 46 to 23. So when a sperm cell from the father fuses with an egg cell from the mother, the result is a single fertilized cell, which again contains 46 chromosomes. Meiosis begins with a cell in the sex organs, in interphase, and closely resembles the beginning of mitosis. It is only when the chromosomes begin to form that we can see a noticeable difference. A remarkable thing begins to happen. Pairs of look-alike chromosomes approach each other and join along their entire length. The chromosomes come so close together that they overlap each other. Genetic material is exchanged during this union between the points at which the chromosomes overlap. The exchange of segments in this twisting and tangling of chromosomes is called crossing over. If we keep in mind the fact that each chromosome can carry thousands or more genes, we can begin to appreciate the staggering number of genes that can be shuffled during crossing over. The pairing of look-alike chromosomes only occurs in meiosis. Crossover can be easily observed in living cells and is one of the most visible differences between meiosis and mitosis. In this salamander cell, crossover is occurring between pairs of look-alike chromosomes. The various figures created by the chromosomes during crossover are called chiasmata. After crossing over, the chromosome pairs partially separate and migrate towards the center of the cell. There, they separate completely. Unlike mitosis, these chromosomes still contain 2 DNA molecules. The cell membrane then pinches in, forming 2 new cells. Each of these new cells now enters a stage that is reminiscent of interphase, except that no new DNA is replicated. The chromosomes again migrate to the center of the cell. The centromeres split and the chromosomes separate, each half moving to the opposite sides of the cell. These new chromosomes only contain 1 molecule of DNA, so that the 4 cells that now form have half the number of chromosomes and half the number of DNA as the body cells of the organism. Let's watch meiosis actually happen in a salamander. As the chromosomes begin to appear, crossing over is occurring. The chromosomes partially separate and then align themselves in the center of the cell. They then separate completely and two new cells form. While in salamanders a nucleus forms and the chromosomes disappear, in some organisms no nucleus is formed and the chromosomes remain visible. Even though this pause strongly resembles interphase, it is important to remember that no new DNA is replicated in this stage of the cell's life cycle. The beginning of the second meiotic division is marked by the reappearance of the chromosomes. In cells about to undergo the second division, the chromosomes migrate directly to the center of the cell in a manner reminiscent of mitosis. Once there, they separate, and the second division soon follows. The 4 cells that result from the 2 divisions of meiosis have half the number of chromosomes and half the amount of DNA as had the original cell previous to interphase. These new cells now develop into sperm, which are able to fuse with an egg cell, restoring the chromosome number to that of the body cells of the organism. During interphase, the cell replicates its DNA. Crossing over begins when look-alike chromosomes pair. After crossover is complete, the pairs partially separate and line up in the center of the cell. Splitting occurs with each chromosome still containing 2 DNA molecules. The genetic composition of these molecules has been changed. 2 new cells are created by the first division, each with DNA still equal in amount to the organism's body cells. The cells enter a resting period during which no DNA is replicated. The chromosomes then reappear and line up. However, no crossover occurs during this time. After splitting, each chromosome has a single molecule of DNA. At the end of the second division, each cell has half the amount of DNA and half the chromosome number as the original cell had before entering interphase. Because mitosis and meiosis appear at first glance to be remarkably similar, let's quickly compare their differences. In both mitosis and meiosis, the cell enters interphase with 4 chromosomes, each of which has 1 DNA molecule. In both cases, the DNA is doubled during interphase, resulting in cells that have the same amount of chromosomes but twice as many DNA molecules. In mitosis, the chromosomes line up in the center of the cell. In meiosis, look-alike chromosomes pair and crossing over occurs. In mitosis, the centromeres split and the new chromosomes contain 1 DNA molecule. In meiosis, the chromosome pairs partially separate and line up in the center of the cell. In mitosis, division occurs. Each of the new cells has the same number of chromosomes and the same amount of DNA as did the original cell. In meiosis, the chromosome pairs separate after they have exchanged genetic information. Each chromosome contains 2 DNA molecules. After division, meiosis continues as the chromosomes migrate to the center of the cell. The centromeres then split and the chromosomes move to opposite sides of the cell. Each chromosome now has only one DNA molecule. Each of the 4 cells formed has half the number of chromosomes and half the amount of DNA as did the original cell.